Welcome back to the Sand Science Experience, where we explore the most exciting new research papers and preprints just for you. You're listening to a deep dive into some fascinating new science. Before we jump in today, we just want to take a quick second. If you could like this video and subscribe to the Sand Science Experience, it really helps support the channel.
It does. It lets us keep bringing you these deep dives into, the latest stuff.
Exactly. Okay, so let's get into it. Today, we are tackling something absolutely fundamental. Mitochondria, those tiny powerhouses inside basically every cell you have.
Right. And specifically, we're zooming in on dysfunction in mitochondrial complex 1.
Now, stick with us because, yeah, it sounds technical, but the real story here, it's surprising, sometimes kind of contradictory relationship with lifespan and aging.
Yeah, we've got some really interesting findings from mice and also from fruit flies. And they offer, well, quite different takes. Makes you rethink things.
It really does. I mean, mitochondrial dysfunction, it's becoming so central to how we think about aging, right?
Yeah.
One of the main hallmarks.
Absolutely. And complex 1, NADH.ubiquinone oxidoreductase. It's crucial. It's this huge complex, the biggest one in the electron transport chain.
That's the machinery making most of our energy, our ATP.
Precisely. Complex 1 is the main doorway for electrons getting into that chain. And it's not just energy. It helps regenerate NAD plus SAE, which is vital for metabolism. And it can also produce reactive oxygen species, ROS.
Which can cause damage, but they're also signaling molecules sometimes.
Exactly. So you see, problems with complex 1 can have these ripple effects all through the cell, the whole organism even.
Okay, so our mission today, take these two recent papers, unpack what they're telling us about complex 1 going wrong, and see how that hits health and longevity.
And we'll look at where they agree, but maybe more importantly, where they really diverge, because that raises some provocative questions about how we even study aging.
Definitely. So let's start with the first paper. We'll spend most of our time here, maybe say 3/4 of the discussion. It's from Geroscience, and it focuses on a specific mouse model.
Right. This is the NDUFS4 knockout mouse, the NDUFS4 gene. You need it to build a working complex 1. So in these mice, the gene's knocked out, that's the double minus sign, and complex 1 is, well, severely deficient.
And these mice, they're actually a major model for a human disease, right? Leigh syndrome.
That's right, Leigh syndrome. It's a really severe pediatric neurodegenerative disorder. Hits infants, causes rapid decline, often fatal within just a few years.
Just awful. And it's caused directly by genetic problems affecting mitochondria, often complex 1 defects.
And the reason aging researchers are interested in a model for a childhood disease like this, well, the paper points it out. Mitochondrial dysfunction is a key part of aging.
Ah, okay. So the problems you see in Leigh syndrome, the brain degeneration, metabolic issues, they kind of overlap with age-related problems.
Exactly. Things like neurodegeneration, metabolic disorders we see in aging. So the paper sort of frames this NDUS4 mouse as a potential test bed, maybe a faster, cheaper way to test interventions that might target those fundamental aging processes.
Interesting idea. Can a model of such a severe early developmental issue really tell us about the slow creep of normal aging? That's a key question kind of hovering here, isn't it?
It is. But first, let's look at what life is like for these mice, according to the paper. It's grim. Lifespan is incredibly short, average around 57 days, median 55 days.
Wow, barely 2 months. Normal lab mice live, what, 2-3 years?
Yeah, exactly. So it's a severely compromised model. And they develop very clear neurological symptoms. One key thing they track is trunk curl, or clasping.
That's when you lift the mouse by the tail and it pulls its legs in.
Right. And this usually shows up pretty predictably, around 41, 42 days old. The paper found a strong link between when clasping started and how long the mouse lived after that. It's a clear marker of the disease getting worse.
And there were differences between males and females. That's always interesting.
Yes, quite fascinating. Even though the clasping started around the same time in both sexes, the female mice seemed more resilient after symptoms began. They lived about 5 days longer than the males once they started clasping.
Well, any idea why?
The paper doesn't fully explain it, but they did note something about weight. There was a negative correlation between body weight at day 30, like postnatal day 30, and lifespan, but only significantly in females. Smaller females at that early point tended to live longer.
So maybe hormones, metabolic differences. It's a common theme in aging research, those sex differences.
It really is. Complex 1 biology itself seems to have sex-specific aspects. Understanding why females hold on longer here is still an open question.
Okay, now for the part that really got the aging field buzzing. Longevity interventions. They tested compounds known to extend life in normal, healthy mice. Stuff from the NIA's Interventions Testing Program, the ITP.
Right, the ITP, that's the big program the National Institute on Aging runs, testing potential anti-aging drugs really rigorously.
And what happened when they gave these to the sick Indus4 mice?
The results were pretty striking. These interventions that work in healthy old mice, they also significantly helped these really sick young Indus4 mice, specifically Rapamycin, Acarbose, and 17-alpha-estradiol. All of them significantly boosted lifespan and delayed the onset of symptoms like clasping.
Wow. Okay, let's break that down. Rapamycin inhibits mTOR, right?
Yeah.
Big pathway in aging.
Yep. Acarbose affects carb digestion, maybe works partly via the gut microbiome.
And 17-alpha-estradiol. That's the steroid that helps male mice live longer in the ITP studies.
That's the one, a non-feminizing form. And seeing all these different types of interventions work in this specific severe mitochondrial disease model, it suggests they're hitting some shared biology, something relevant to both this disease and normal aging.
So it's not like they're fixing the broken complex 1 gene.
No, they can't do that. The idea is they're maybe boosting overall resilience or targeting downstream pathways that go wrong in both scenarios. The authors even float the idea that these drugs might be working systemically.
Like affected the gut or the liver. And that helps, even though the main symptoms look neurological.
Potentially, yeah. It suggests the effects aren't purely brain-focused, even if the disease manifests strongly there.
This actually led the authors to propose something pretty bold in their discussion section, didn't it?
They did. A hypothesis that aging itself could be seen as essentially a chronic acquired mitochondrial disease.
Whoa. That's provocative. Like, the slow decline of aging is just accumulated mitochondrial damage, a milder version of Leigh syndrome.
That's the implication. It reframes aging quite dramatically.
Did they look at anything else? Environmental factors?
They did check temperature, housed some mice at thermoneutral temp, about 30 Celsius. That's usually easier on mice, less energy needed to stay warm compared to standard lab temps.
And did it help these Indus4 mice?
Nope. Didn't affect survival or disease progression, which suggests maybe the problems these mice have with staying warm are secondary, a consequence of the main disease, not something you can just fix by making them warmer.
Interesting detail. And one last thing for the mouse paper, they mentioned NAD plus precursors, like NMN or NR. Those get talked about a lot.
Right. They tried them, but in their hands, under their specific conditions, they didn't see significant effects on survival or symptoms. They do note this is different from some other reports using those compounds in this model.
So it highlights how tricky these experiments can be, sensitive to the details.
Exactly. A good reminder that results can vary. Things aren't always perfectly consistent across different labs or setups.
Okay, so that first paper paints a picture. Severe early complex 1 problems look a bit like fast-forwarded aging, and crucially, aging interventions work. It supports using this model as a test bed. But now, prepare for a plot twist.
Right. Let's switch gears to the second paper from MBO Reports. This one uses Drosophila, fruit flies, and it brings in a totally different angle, timing.
Timing? How so?
This fly study is really cool because it asks, when does the mitochondrial problem start? They set it up so they could mess with complex 1 at different life stages.
Ah, okay. So some flies had bad complex 1 from early on, like the knockout mice.
Exactly, from development. And then other groups, they only induced the complex 1 problem after the flies became adults.
And what did they find? Was it the same outcome?
Not at all. This is the really striking part. The flies where complex 1 was depleted during development, they grew up to be adults with a severely shortened lifespan. And they were way more sensitive to stress, oxidative heat, starvation. Kind of what you expect, right? Mess up energy early, get sick adults.
Makes sense so far. What about the other group, the ones where complex 1 was only hit in adulthood?
This is the bombshell. Those flies, they did not have a significantly shorter lifespan or reduced stress resistance.
Wait, really? But did they actually have lower complex 1 activity as adults?
Yes, they checked. The adult flies from both groups, the developmental onset and the adult onset, had comparable reductions in complex 1 function. But one group died young and frail, the other lived basically normally.
Wow, okay. So it's not just how much complex 1 activity you have as an adult.
Doesn't look like it. The paper proposes it's all about the timing. Hitting complex 1 during that really plastic-sensitive developmental stage, it triggers what they call a maladaptive response.
Maladaptive. Like the fly tries to cope, but messes things up for later life.
That's the idea. They used multi-omics, looking at genes, metabolites, all sorts of data, and found metabolism was dysregulated, the fat body was wasting away. but only in the flies where the problem started early. It's like the developing fly gets wired wrong in response to the early defect.
But if the hit comes only in adulthood, the system is maybe less plastic. It doesn't trigger that same damaging cascade.
That seems to be the hypothesis. The adult system responds differently, or maybe just can't launch that specific maladaptive program.
And this, the fly paper authors say, helps make sense of conflicting results from earlier studies.
Yes, because there were reports going both ways with complex 1 in invertebrates. Sometimes knocking it down shortened life, sometimes like certain gene tweaks in C. elegans worms done developmentally, it actually extended life. The fly data really suggests that developmental context is king. It shapes the whole adult outcome.
Okay, let's connect this back to the mouse then. If timing is so critical in flies, developmental hit causes severe disease, adult hit doesn't shorten life. What does that say about the Inducible mouse model for studying aging?
That's the million-dollar question, isn't it? The Inducible mouse has the defect from day one. It causes severe early disease. How well does that truly model normative aging, which is mostly about decline happening much later in life?
Yeah, is it really a model of aging, or is it primarily a model of a specific devastating mitochondrial disease state that happens to start early?
It's a crucial distinction, and if it's more of a disease model, are those aging interventions working in the mouse because they're fixing those specific maladaptive developmental programs that got set in motion early on,
rather than fixing the mechanisms of slow, late-life decline?
Potentially. The fly data really makes you pause and question how directly we can translate findings from these severe early onset models to the process of normal aging. The organism's response might just be fundamentally different depending on when the insult happens.
It definitely complicates the interpretation of the mouse results. Now, we should add the caveat the fly paper authors included, right?
Yes, absolutely crucial. While their results are super suggestive, they are careful to note that the findings, especially around the adult-only induction not affecting lifespan, need more direct experimental proof within Drosophila. They mention some technical challenges in doing the ideal follow-up experiment, like restoring complex 1 in adulthood after it was knocked down developmentally.
So the evidence points strongly towards timing, but some specific conclusions need firming up.
Exactly. Science in action. One study opens big doors, but also shows where the next steps need to be taken.
Okay, so let's try and synthesize this. We have two papers, both showing complex 1 is critical. The mouse study shows a severe early defect causes early death, but importantly, standard longevity drugs help. Supports it as a test bed, suggests shared pathways with aging.
But then the fly study throws a curveball, shows a dramatic dependence on when the dysfunction hits. Developmental hit. Bad news for the adult. But an adult-only hit, even to the same level. Surprisingly, lifespan is okay, likely due to avoiding those early maladaptive responses.
Which really makes you think complex 1 dysfunction isn't one single thing. The context, especially the developmental stage, seems to change the outcome dramatically.
Yeah, and the fly results really challenge the idea of using these severe developmental disease models as perfect proxies for late-life aging. Are the interventions in the Inducible mouse fixing the early programming error, or are they boosting general resilience that helps in both situations?
It's probably tangled up, but the fly work forces us to be more precise, right? Distinguish developmental problems from adult onset changes.
Definitely complicates things.
And this leads us to a final kind of provocative thought for you, the listener, to chew on. Given these findings, especially the fly data, where adult-only complex 1 decline didn't shorten lifespan, and knowing that complex 1 activity does tend to decline naturally with age in flies, and humans too.
Where are you going with this?
Could that natural late-life decline in complex 1 actually be protective? An adaptation against aging rather than a cause of it?
Whoa. Okay, so instead of seeing it as purely detrimental decay, maybe the cell deliberately dials down complex 1 in old age as a way to cope with other stresses.
Exactly. Maybe that downregulation is adaptive. Unlike the system's disastrous reaction when complex 1 is hit hard during development, it completely flips the script.
That is provocative, suggesting an age-related decline could actually be beneficial. Very different from the usual narrative.
Lots to think about for sure. We've really only scratched the surface of what these papers imply.
Absolutely. The links between mitochondria, development, aging, it's clearly way more nuanced than just mitochondria fail as we get old.
Well, that wraps up this deep dive on complex 1. Thanks so much for joining us. And please remember to like this video and subscribe to the Science Experience.
Yeah, stay tuned for more deep dives into the science.